Welcome viewers, greetings. In today's class, we are going to see about human influenza virus. So the virus belongs to the family Orthomyxoviridae. Orthomyxoviridae comprises of RNA viruses that includes 5 genera: influenza virus type A, influenza virus type B, influenza virus type C, Isa virus and Togoto virus. Influenza virus causes influenza in vertebrates including birds, humans, and other mammals. Isa virus infects salmon fish. Togato virus infects vertebrates and invertebrates such as mosquitoes and sea lice. General characteristics of Orthomyxoviridae includes they contain negative sense single-stranded RNA viruses, pleomorphic in its existence. It means it can change its size and shape irregularly. So it mostly spherical in shape, sometimes tubular in shape. Size ranges between 80 to 120 nanometer. This is the structure of the viruses, which are pleomorphic and changing its shape at any given time. Structure contains an outer envelope that contains two glycoproteins. One is hemagglutinin HA and neuraminidase NA. Hemagglutinin facilitates clumping of the human RBCs, whereas neuraminidase facilitates the entry of the virus into the host cell. It has a matrix protein M1 and it has membrane proteins also that gives its shape. Type A and type B viruses contains 8 different helically coiled segments of RNA along with nucleocapsid proteins, whereas type C influenza contains 7 genomic segments of RNA. And this is the structure of the virus. Here hemagglutinin are labeled as H spikes, whereas N spikes are the neuraminases. It has an outer envelope made up of lipid, and there is a matrix protein inner to the lipid layer, and it has a nucleocapsid that is surrounding the helically coiled RNA. And there are some antigenic shifts. So the hemagglutinin and neuraminases of influenza A virus can undergo major and minor antigenic changes to ensure the presence of immunologically naive, that is the susceptible people. For the susceptible people, shift occurs only with the influenza A virus, and the different hemagglutinins are generally found in the as a result of the antigenic shift and are designated between H1 to H16. Neuraminases of influenza A also undergoes antigenic shifts and results in different types of N variants. So mutation derived changes in hemagglutinin are responsible for the changes in antigenic shifts. In influenza type B virus, it undergoes only minor antigenic changes. Hence, influenza A is highly infectious because of the antigenic shifts. It can evade immunity and can evolve into new type of viruses. So this process occurs at every 2 to 3 years, causing local outbreaks of influenza A and B infection. And while discussing the pathogenesis, we have to concentrate on transmission, replication, and mechanism of the disease incidents. So in transmission, it is transmission mainly between the person to person by just sneezing, coughing, and simple talking. Virus likes a cool, less humid atmosphere for its multiplication and survival, which is winter heating season is more suitable. So it extensively spread by the school children when they are placed in a closed atmosphere. The respiratory tract is the gate opening of the virus. So influenza can also be transmitted by saliva, nasal secretions, fecus, and also through blood transmission. It can be easily transmitted and harder to fight. And if you look into the replication begins with the binding of hemagglutinin to the sialic acid receptors on the cell surface glycoproteins. So sialic acid residues are found on the cell surface glycoprotein of the host cell. So hemagglutinin goes for an interaction with the sialic acid residue, which facilitates the attachment process of the virus, and then the virus is then transferred to the endosomal vesicle with the help of neuraminidase. It is cleaved. The receptor as well as the hemagglutinin contact is cleaved with the help of neuraminidases. So the nucleic acid travels to the nucleus where it is transcribed into the messenger RNA, and this messenger RNA is the translatable RNA. So transcriptase uses the host cell. Transcriptase uses the host cell mRNA as a primer for viral mRNA synthesis. So the mRNA are translated into protein within the host cell cytoplasm. The hemagglutinin and neuraminidase glycoproteins are processed by endoplasmic reticulum and Golgi apparatus of the host cell. Once it is processed and produced, hemagglutinin and neuraminases are transported to the cell membrane by the fusion of the endoplasmic reticulum and Golgi apparatus with that of the host membrane. Then the virus buds selectively from the apical surface area of the cell. We call it as airways area, especially the nasal tract, as a result of the preferential insertion of the hemagglutinin in the host membrane. So virus is released approximately 8 hours after the infection to infect new individuals. And this is the replication cycle, where the first is the attachment or binding process, where hemagglutinin of virus interacts with sialic acid residues found on the glycoprotein of the host cell receptor. Then after attachment it goes for entry into the endosomal vesicle. Then lysosome as well as endosome fusion happens to form phagolysosome. So within the phagolysosome, the entire outer covering of the virion get digested, and the 8 helically coiled nucleic acid is released into the cytosol, host cell cytosol. These ones are negative sense single stranded RNA that enters into the nucleus, and where it goes for a simple transcription in the third step, and transcription into positive sense RNA with the help of host cell RNA primers. And these positive sense RNA go for multiple replication, and then it will go for expression into protein, like as it is it's a eukaryotic host cell. See, the mRNA capping and poly A tail addition will be there in the viral RNA. All will enter into ribosomes that are studded onto the endoplasmic reticulum, where the viral proteins are synthesized, especially hemagglutinin and neuraminidases. As these are glycoprotein, their glycosylation will be facilitated by the Golgi apparatus, and these hemagglutinin and neuraminases will be transported towards the membrane, where it get embedded into the membrane. And this localized site is ideal for the synthesis of negative sense RNA, are the sites for budding, and the positive sense RNA are again converted into the negative sense mother template, their progeny templates, and these ones will assemble in the given area where hemagglutinin and neuraminases are assembled, and through a simple budding process, new virions are released from the host cell. So this particular image is brought from Murray et al. Medical Microbiology 7th edition. Important product of influenza gene segment. So if you consider 4 segments, the 4th segment, it codes for the protein hemagglutinin. Hemagglutinin interacts with human RBC and causes clumping, and hence the name given as hemagglutinin. It is the viral attachment protein, and this 4th segment of the RNA codes for hemagglutinin fusion protein, and it is the target of neutralizing antibodies. So proteins like hemagglutinin are generally targeted by the antibodies of our own immune system to neutralize the virus. And segment 5 that codes for nucleocapsid, so capsid proteins that cover the RNA. And segment 6 that codes for neuraminidases, which only cleaves the sialic acid as well as the hemagglutinin bound complexes, and it promotes the viral entry, or we can call it as viral release from the phagosome. It facilitates the uncoating process, that is the step 2 in the viral replication. And there is segment 7 that codes for two protein, that is matrix protein M1 and membrane protein M2. Matrix protein is the viral structural protein. It interacts with the nucleic acid as well as the envelope and promotes the assembly of the entire virus in the release process. In membrane protein, here it forms membrane channel and target, and these are all the targets for the drugs, antiviral drugs like amantadine and rimantadine. It facilitates the uncoating process. Membrane protein facilitates the uncoating process and hemagglutinin production. So pathogenesis, generally we divide it into the asymptomatic period and symptomatic period. In asymptomatic period, that we call it as incubation period, it proceeds for 1 to 4 days and an average 2 days. So adults can be infectious from day 1 before the start of the symptoms to the 5th to 7th day after the illness that starts. It is more infectious in children. So the main cause of hospitalization due to flu is by influenza virus. The symptoms and the time course of the disease are determined by the extent of viral and the immune killing of the epithelial tissue and cytokine action. Influenza is normally a self-limited disease that rarely involves organ damage other than lungs. So repair of the compromised tissue is initiated within 3 to 5 days of the start of the symptoms, but may take as long as a month or more, especially in the elderly people. So here is the pathogenesis. If you look at the inoculation of the virus into the respiratory tract on the day 0, incubation proceeds for about 2 to 5 days, and vague symptoms of influenza starts. It is the symptomatic influenza-like disease. And what happens? Virus in the respiratory secretions we see between 5 to 7 days, and the people are actually infectious. They are spreading the virus to the healthy individual before they get the actual severe symptoms. And from day 7 onwards, here there is a rise in the virus specific antibody in the serum of the individual. Here also, due to the lack of immunity, there can be secondary bacterial infection from the day 7 onwards, and there is also a cytokine rise, induction of the interferon, that is the antiviral immunity from the day 2 onwards. And there can be pathological changes that are actually evident in the respiratory tract, that is the damage of the respiratory tract mucosa that starts from day 8, and there is initiation of the cell mediated immunity from the day 5 onwards, and then there is a long convalescence period up to 26 days, and the disease subsides. So the image is brought from Murray book on medical microbiology, 7th edition. So influenza initially establishes a local upper respiratory tract infection. To do so, the virus first targets and kills the mucus secreting ciliated epithelial cells, causing the loss of this primary defense mechanism. It actually attacks the innate immune component, the anatomical barrier. Then neuraminidase facilitates the development of the infection just by cleaving the sialic acid to hemagglutinin contact. So sialic acid residues of the mucus, neuraminidase cleaves the sialic acid residues of the cells on the respiratory tract, thereby providing access for the virus to enter into the tissue. So preferential release of the virus at the apical surface of epithelial cells and into the lung promotes cell-to-cell spread and transmission to other hosts. If the virus spreads to lower respiratory tract, the infection can cause severe desquamation, like the epithelium will go for cleavage and shedding of the virus. So if the virus actually spreads to the lower respiratory tract, that is trachea and then lungs, the infection can cause severe desquamation or shedding of the bronchial or alveolar epithelium down to the single cell basal layer or to the basement membrane. Pathogenesis. What else does influenza harm then? Yeah, influenza infection promotes bacterial adhesion to the epithelial cell. As a result, there can be secondary bacterial infection like pneumonia may result from the viral pathogenesis, or from a secondary bacterial infection like streptococcus pneumoniae can also be possible. So inflammatory cell response of mucosal membrane. On the mucosal membrane, we can see a recruitment of monocytes, lymphocytes, and few neutrophils. Submucosal edema, there is thickening of the submucosa, and there can be hyaline membrane disease, alveolar emphysema, and there can be necrosis of the alveolar walls, cell death of the respiratory alveolar walls. So, major contribution of pathogenesis of the influenza syndrome, immune or marked with violet arrow, purple arrow, solid line arrow for the immune response, and dotted line arrow that shows less frequent outcomes. So first, aerosol inoculation of the virus into the respiratory tract, replication of the virus in the respiratory tract, followed by desquamation of the mucus secreting ciliated epithelial cells, and that results in influenza-like syndrome. And what happens between the replication and desquamation? There is a rise in the interferon, that is the antiviral cytokine, and this is followed by rise in the antibody and T cell mediated immune response. So, rise in the antibody and T cell mediated immune response always gives a future protection, a long term protection against influenza virus. Interferon rise as well as T cell response prevents the influenza syndrome from desquamation to the syndrome phase. And in addition, the secondary bacterial infection can also result once the mucosal membrane as well as lower respiratory tract is desquamated. There can be pneumonia like symptom. Primary viral pneumonia can result. If the virus spreads into other parts, there can be secondary infections in the central nervous system, as well as muscle fibers can also be damaged. So, what are the symptoms? Symptoms can be simply given with the term facts. First is fever and ache, probably headache to any body ache, and then chills, shivering chills, C for chills, T for tiredness, and then S for sudden onset of the symptoms. So, fever, aches, chills, tiredness, and then sudden onset are the 5 major pathognomonic symptoms of influenza virus. A brief prodrome of malaise and headache lasting a few hours. Okay, so first initially we will get the tiredness and muscle pain, headache for few hours, then onset of the facts: fever, chills, severe myalgia, severe muscle pain, loss of appetite, loss in the hungry. So loss of appetite, weakness, fatigue means feeling tired, and sore throat, and there is a non-productive cough, which means a dry cough. So fever, chills, muscle pain, loss of appetite, weakness, tiredness, sore throat, shows on the throat, and then there is a dry cough, and there is recovery within 7 to 10 days for normal healthy individuals. In case of immunocompromised individuals, there can be secondary infections like pneumonia, myositis, and then Ray's syndrome can also be possible. So, symptoms of influenza: if suppose it is the central symptom, then there can be headache. If it is systemic, you will have usually high grade fever. Then in case of nasopharynx, if the virus replicates, then there is a runny nose and stuffy nose, sore throat, and aches. And if it enters into the secondary sites like muscle, at extreme cases, you will have the myalgia and fatigue, tiredness, muscle pain. And in the lungs, it shows coughing and respiratory syndromes, and in the gastric tract it induces vomiting, and in the joints it causes rheumatism, it causes pain, joint pain. So, and then epidemiological distribution, that is also seasonal, especially in India. It is all over the year, especially in the southern part of India, western part, and the eastern part of India. It is all over the year, as it is colored yellow. And in the South Africa, South America, and South Australia, it is season between April to November. Its peak is between April to November, and between November to April, because of the climatological factors, the incidence of influenza is high in northern part of America, Russia, and the northern part of Africa, as well as Finland, Europe, and other countries. So it is also sporadic, and it is also sometimes in some tropical countries. It is all over the year it is found. Typically, in a year, normally 2 flu seasons can happen, one per hemisphere. There are between 3 and 5 million cases of severe illness, and around 5 lakh deaths worldwide is occurring due to influenza viral infection. Roughly 3 times per century, a pandemic can also occur. A pandemic has occurred 3 times per century, which infects a large population of the world and can kill 10 million people. 10 million people. Based on the characteristic symptoms, doctors generally diagnose based on the objective and subjective symptoms, and also the season, presence of the virus in a given community. We can diagnose the disease, and we can go for cell culturing of the virus on the monkey kidney cell line. We can go for testing the heme agglutination, because the virus carries the heme agglutinin, and there can be antibody inhibition through heme adsorption based assays, or heme agglutination inhibition assays can also be performed to diagnose the presence of the active virion in the bloodstream. Prevention is through a single shot containing the killed virus or inactivated virus of vaccine can be given in the arm normally, can also be given as a nasal spray. So vaccines are not frequent. In severe cases, we need to go for vaccine administration. A single shot of the killed virus or inactivated virus in the arm, right or left arm. And we have to maintain other control measures, like you have to have a good hygiene. You have to cover the nose and sneeze when you sneeze, and you have to stay away from the sick people. You have to follow the sanitary procedures to avoid the viral incidents. And treatment, we have acetaminophen, antihistamine, and other drugs to relieve from the influenza-like symptoms that looks like allergic symptoms that look very similar to the allergic symptoms. So we need antihistamines and acetaminophen. And we have antiviral drugs like amantadine and rimetadine that inhibits the uncoating step of the influenza A virus, but do not affect influenza B and type C. Zanamivir and oseltamivir inhibit both A and B as enzyme inhibitor of the neuraminases. As a result, the uncoating of the virus can be prevented. So thank you so much for your patience.